modeling a research topic. This is a program that is run out of the Milwaukee School of Engineering Center for Biomolecular Modeling, where we capitalize on our 3D printing technology in a way to engage students in understanding molecular biology. So SMART Teams began about 12 years ago with a group of high school teachers who were very interested in learning about the technology, this 3D printing technology. They explored a topic. They actually looked at ribosomes, and they were able to talk with Tom Stites, a Nobel laureate from here in Wisconsin, who won the prize a few years ago. And so they were very excited about this opportunity to work with the researcher to explore a specific topic in depth, and then to create a 3D physical model of that particular protein. So these 6 teachers, super excited about it, said we have to take this to the classroom. How do we do it? And so we developed the Martine Program. So in 2001, we had our very first SMART team, and this was out of the Riverside University High School. These 3 students worked with their researcher, Dr. Tang, and they explored proteins related to anthrax. So if you remember back to fall of 2001, this was when 9/11 happened, and then shortly thereafter we had anthrax being sent through the postal system. So this was an opportunity for these students to explore a very timely, pertinent topic. In fact, so timely that these students created the first ever physical models of a protein involved in anthrax that had just been determined. So it was very topical. These students also had a chance to send their models to a congressional hearing in Washington, D.C. So mind you, again, this was right after 9/11. Everything, people were very sensitive and suspicious of things happening. And we sent these models in little boxes that were labeled anthrax. People walked right into the congressional hearing without any problem. Let's also add that our printer works with white powder plaster in order to generate the physical models. So the fact that these models even made it into the congressional hearing is amazing to me. But they were really well received, and so very excited. So from our very first SMART team in 2001, we now have over 60 SMART teams across the U.S., and I just apologize, my map doesn't show up quite as well. So pretend that you know where all your states are. And all of the little stars represent SMART teams that we have across the U.S. So it's exciting for us. We have about 20 that we work with here in Milwaukee every year, and we have an additional 40 that are happening across the U.S. And each spring we have the opportunity to go to a national research meeting, and a subset of these SMART teams, actually 25 of them this year, will be landing in Boston, and we're going to have a chance to actually have a representation at this meeting. So it's very exciting to go from that one team in 2001 up to 60 of them now. So through the support of grants from the National Institutes of Health, also the Howard Hughes Medical Institution.
I would not have this awesome new job if it wasn't for an awful lot of people. Students, first of all, let's give yourselves a hand, because it's been a long 6 months. In addition to that, you've had a teacher advisor who has guided you along the way as well. These teachers have played with us at the CBM during the summer. They've taken a course that allows them to host a SMART team. And so we'd like to recognize this afternoon's teacher advisors. So when I announce your name in school, please stand and stay standing. From Kettle, Moraine High School, we have Stephen Plum. From Cedar High School, we have Karen Tiffany. This program cannot run without the volunteer mentors that we have at several of the universities and research facilities in the area. So our heartfelt thanks go out to all of you guys who do bench work, teach classes, and carve out time to spend with our high school students and their teacher advisors. Again, thank you so very much. We'd like to recognize you as well. So when your name is called in your institution, please stand and remain standing. His mentor was Anil Chawla. He is from the Medical College of Wisconsin. Okay. Cedarburg High School, their mentors were Audra Kramer and Nishat Gurgas from the Medical College of Wisconsin.
So part of the SMART Team program process is we have to set out goals and objectives of what we hope to accomplish during the year. And so this summer we took a very close look at what we were looking to do within the SMART Team program. And our main purpose, or what we strive to achieve, is to introduce the SMART teams to the community of science. So if you think about science in the sense of, researchers doing their work, they go to conferences, they publish papers, they collaborate, they do a lot of interaction. And if you're over here in that science world, it's a good world. You know what you're doing, you know how it happens. If you're a student on the outside, it's very hard to break into that. And so what we hope to achieve with SMART teams is to kind of bridge that gap a little bit and start to introduce students to this community. And we do this by a number of different things. One of the first things we ask our students to do is to be able to discuss how modeling is a practice of science. So researchers have in their head an image of what they think is happening, and it's great to see all the works and they can write it down on a piece of paper and draw out pictures, but being able to communicate that over to students is a challenge that all of us face. So we want students to really recognize how modeling is used within the field of science. And for our high school students, the next generation science standards that will be adopted here very shortly really do stress that importance of modeling as a scientific practice. We also hope that the students are able to explain their story related to the protein structure and function. So every protein that's out there has a specific shape, and with its specific shape comes a specific function. So we work together to explore that structure-function relationship, and more importantly, within the context of a big picture. So how does this work in my world? Why do I care about this protein? I think I've asked that question of our SMART teams 1,000 times this year. Why do I care? Make me want to care. Every protein has a great story. Get me excited about it. So hopefully today you'll get excited about some of the proteins that they've been working on. We also want them to be able to look at experimental evidence and be able to apply that evidence in a way that supports their story. So researchers are in their labs, they're asking questions, they're collecting data, they're putting the data together to say, this is how it works, here's my model of what's going on. And so we want the students to be able to take some evidence, some of the data, and be able to explain how that supports their story. We also want students to be able to understand that there is this community out there and how these community members all interact with one another. A lot of times it is a very collaborative nature in the sense that we work well together and we can share stories, and we can feed off of each other. Sometimes there's an element of competitiveness, and so we want to also introduce that aspect as well. So as these peripheral participants learning about the community, we want them to be able to demonstrate their ability to become members of that scientific community by presenting their story, either in a poster or in an oral presentation format. Our posters are designed to be towards a scientific audience in the sense that you're communicating your story to colleagues who are familiar with your particular story or general subject matter. And then we also have this oral presentation day for students to practice presenting their stories to a general audience where that biochemistry background may not be up to the same level as talking with people who have been researching for 30 years. So we divided our SMART team program into 3 different phases. There's the qualification phase, the research and design phase, and the presentation phase. During the qualification phase, we have all of our SMART teams come to the Milwaukee School of Engineering several Saturdays in the fall. And we start the first Saturday with explaining different properties of basic protein biochemistry. So water, amino acid structure, protein structure, and also about writing abstracts. We do this all on a very early Saturday morning, so usually from 9 to noon. And as my pictures fly by for the remainder of my talk, I want to point out all the smiling faces, so even though we have them there early Saturday, they're engaged. See? So the CBM really works to develop hands-on materials to get students excited about learning protein biochemistry. So part of the reason why we can have smiling faces early on a Saturday morning is that we get the kids up and moving and doing the models and working with the activities. And so it gives them an opportunity to have some fun, but also to learn some protein biochemistry. And so this first Saturday, we really focus on understanding how proteins fold and what that final shape is going to be. We also do spend some time talking about writing abstracts and how do you communicate your science in a very short, finite little bit of word space. So I usually give them about 200 words that they can play with in order to write that abstract. 200 words sounds really great, it's not a 10-page paper, until you actually sit down to write the 200 words, and then it becomes a struggle. And we go back and forth several times, and most students usually get tired of seeing the email, yeah, that was great, you're really making good progress. You need to still work on this. And so we go back and forth a bunch of times, and hopefully honing it down to a concise 200 words. Our second Saturday, we get together here at the medical college, and we use their computer lab to start to explore how do you use the computer visualization program, JMOL. How do we take data of an X-ray crystal structure determining the shape of our protein, put it into a computer environment where we can selectively say, I want this part to be green or this part to be red, and then that eventually translates into a 3D physical model. During this Saturday session, the students are also given their qualification challenge. This is a specific protein that we have all of the students investigate. They read some literature articles, and they write an abstract, and they design a physical model. And this year it pertains to bee venom and hyaluronidase, which is why the bee is on all of your abstract booklets. So that's kind of a reference back to our qualification challenge. So after they've done their model design and their abstract, they come into MSOE again and we do a qualification exam. So now you have to prove to me you know what you're doing before I'm ready to match you up with a researcher. So we have a series of questions that we ask the students. We get them exploring protein chemistry again, doing a bunch of activities, just again to demonstrate to me that they know what they're talking about. They get water molecules for all the correct questions that they answer, and so you can see them all having a collection of water around their tables. We do this mostly just to make sure that they're ready to go on. I ask a lot of questions, but I also do kind of pepper in some questions that pertain to things like, the Packer score from the previous weekend, or whatever book series I happen to be reading that I assume everybody else is reading with me. And so it gives them an opportunity to have some fun, to explore the world of science. Again, going back to my, smiling faces. So my question for you this afternoon. Hunger Games. A little bit of a theme this year with our Hunger Games. So after they've shown that they know what they're talking about, we will match them up with their researchers. So the first meeting of our research and design phase is when we have the students come in and we do a little speed dating session. So imagine a room full of tables, 21 tables for all of our SMART teams. Students sit at the tables and our mentors rotate around the different tables, and they get an opportunity to talk to each SMART team for about 3 to 4 minutes. Their jobs are to find out a lot about each other. So the students about the mentors and then the mentors about the students. We have a little quiz at the end to see who learned more, the mentors or the students. But the whole point here is just to show students that researchers have lives outside of the lab. They're not all stuck and chained to the lab 24/7. They have lives. They have favorite books, favorite movies, favorite vacation destinations. We've had mentors who have climbed the Himalayas and gone scuba diving in the Caribbean. So we have a wide variety of activities. Following the speed dating portion and our little quiz, we then match all of our researchers with their SMART teams. And then this begins the conversation about whatever topic the researcher wants the students to investigate. So during this part of the research and design phase, the students work very closely with the researcher to learn the story. They write another abstract, and they design a model. And then that model is then used to tell the story. So our molecular story of what's happening. So in January, we kick off our presentation phase, and this is when students learn about what a poster is, what an oral presentation is, and what we expect them to accomplish over the next couple of months. So we talk about all sorts of things pertaining to posters, what's good about them, what's bad about them. You see all of our poster layouts and different designs. And it also gives them another chance to talk with their researcher to continue that conversation. And now they can use their model as a tool to help facilitate that conversation. It's been a lot of fun over the years to see our researchers get a hold of our models, especially new first-year researchers. They get the model in their hands and suddenly eye contact, gone. Because they're so focused on the model, they're not paying attention to what's going on. They'll still talk to you, but they're still focused on this model and they're kind of looking around. So during these sessions here, you can see our researchers are working very closely with our students in order to generate that poster product. And so on March 1st, we had our poster session down the hall. And this was an opportunity for students to communicate their project to a scientific audience. So if you were at our poster session a couple weeks ago, you know that we set it up where here's the basic science research building and here's the parking lot, poster session right in the middle. Our researchers cannot pass poster sessions without stopping and talking to people. I think that's some gene within people who are in the research field. So it gives them a chance to communicate their science and their project to the scientific audience. And they have their poster, they have their models in hand, and they communicate what's happening at their molecular level. So they talk about the big picture, why is it important, why do I care about that protein, and then they work their way down to a very specific amino acid and what that function is in terms of that big picture. So this was their opportunity to present to a scientific audience. This now, today, is their opportunity to talk to a general audience. So this is their world in which they have to communicate their science to a very general audience. Researchers are often challenged with this because they're used to talking to their colleagues, and their colleagues know what they're referring to as they talk about their specific project. But when we take them out of their colleagues' world and put them into a general audience or a public audience, then it becomes a little more challenging because it's usually I want to jump right in and give you all the specific details. So we've been working with the Martines for the last couple of weeks and getting them to think very big picture and broad ideas about what is so exciting and fun about their particular project. So without further ado, I'm going to turn this over to our SMART teams.
We are the Kettle Moraine SMART team, and we are investigating the role of Gsk3 in the WNT pathway. The WNT pathway is a very involved pathway, and here's a diagram of it in both its on and its off states. In the off state, which is this right here, Gsk3 acts as an inhibitor molecule with these other molecules to block beta-catenin, which is a signal molecule that comes in, and instead of going into the nucleus, it is stopped and it is degraded, which is sort of like how here the Gsk3 is incapable of getting the proper signal through. However, in the on state, the Gsk3 is blocked when WNT binds frizzled on the outside of the cell, which means that beta-catenin is allowed to enter the nucleus and engage in transcription factors. So this is like how Tom is able to properly juggle. This pathway is very important in many cell functions. In particular, healthy cell replication is important, which is like how Jenna and Rachel are able to correctly replicate a pattern with bongos. In addition, this pathway is also involved in embryonic development and making sure structures of the body properly form, such as in cephalization, which is the formation of the head. So Tom, because he has proper cephalization, is able to play trumpet. Also, recent studies have shown that there may be a link between the WNT pathway and neurological health as well. So, Matt here has his pathway functioning normally and is thus able to correctly draw it because he has the mental capabilities to correctly hold and use his drawing utensil. Now, there is a balance between the on and the off states that must be found in order for the cell to function properly. Now, here sort of represents the just right effect, which is how Tom is able to be satisfied by just having the one nutty bar and be properly satisfied by it. However, there can be too much of a good thing. In this case, Tom has had too many nutty bars and is now bloated and improperly satisfied. Or too little activation can be bad as well. Here, Tom has no nutty bars, thus he is left hungry and completely unsatisfied. Here's the model of the Gsk3 molecule. As you can sort of notice, it has roughly 2 symmetrical subunits, which carry out the same effect. In particular, here's the active site of the molecule, where this yellow thing right here, it is normally ATP, which when it is accepted by Gsk3, the Gsk3 molecule is allowed to function properly. In this binding site, how it works is we have these orange molecules over here, which are responsible for latching onto the adenosine part of the ATP. And then you have this red molecule, which binds to the sugar backbone of the ATP. And then these pink molecules that you see scattered through here work using various water molecules and magnesium ions to grab onto the 3 phosphates of the ATP and phosphorylate them. However, there have been several inhibitors to Gsk3 that have been discovered. The more important ones are lithium chloride, and how lithium chloride works is that when it breaks up into the lithium and chlorine ions, the lithium ions end up replacing the magnesium ions found in the active site of Gsk3. Thus, it becomes unfunctional. Now, AMP, which is represented by this diagram over here, is an analog of ATP. The major difference between the two is right here, there's a nitrogen where there should be an oxygen between the second and third phosphates, which means that AMP cannot be phosphorylated the same as ATP. Now, in order to get a better understanding of how this pathway works, we ran our own experiments using zebrafish provided by our mentor, Dr. Anil Shala, and the Medical College of Wisconsin. So what we did is, although there are many experiments that have been done using similar inhibitors, what we decided to do was to do tests using small concentrations of lithium chloride to see what effect it would have on the zebrafish embryo's development. And although we did not get very conclusive results on the much smaller concentrations, at about 10 millimolar, we did notice changes in the development of the embryos. The major things that we noticed were there was much shorter, it was a much slower time period, so the embryos did not develop nearly as fast. We also noticed that there was a lack of pigmentation in the ones with the higher concentrations of lithium chloride. So right here we have the high exposure ones, and they are supposed to be much more darkly pigmented, and they're not. Also, we did notice that the eyes started developing earlier than the rest of the body. So the eyes, earlier on, they are much larger, but then the rest of the body eventually caught up. And also we did get some defects like this here where the body is improperly shaped. So that is a noticeable effect. However, in much higher concentrations, not like how we ran our experiment, there have been much more noticeable effects on the embryos, such as this is a much higher concentration and the embryo has had a failure of cephalization, which means that it has not developed a head, which means that this embryo is not viable. Now, there are several risks in disrupting the Gsk3 and WNT pathway. First of all is the failure of cephalization, which is how Tom he plays his trumpet normally. And then lithium chloride inhibits Gsk3, and the pathway becomes disrupted and does not work correctly. Also, in development, there can be other effects, but also in cancer, certain types of cancer have been noticed to have strange effects with inhibitors such as lithium chloride. For example, colon cancer and breast cancer have had strong effects, so the cells no longer divide normally and thus are out of control, which causes a tumor to form. Also, there have been some suggestions that neurological diseases can be aggravated by disruption to the WNT pathway. So the pathway now she lacks the motor skills and neurological functions required to properly draw a cube. However, there can be several benefits to disrupting Gsk3 as well. For example, certain cancers can be treated by using lithium chloride in order to block the cell division, so the tumor grows much more slowly or stops growing. Also, certain neurological diseases like bipolar disorder and Parkinson's can be treated using lithium chloride to help normalize the pathway. Also, other diseases like diabetes have been found to be affected by the WNT pathway when it's affected by lithium chloride. So in summary, life is a balance between the on and off state of Gsk3 and the WNT pathway. This is known in the science realm as homeostasis, where you have both the on and the off occurring at roughly the same time. And while the WNT pathway is very ubiquitous, it is involved in several things, so disruption can be either very beneficial or very bad in normal cell activity. And in the end, there needs to be a Goldilocks effect where you can't have too much, you can't have too little, you just need to have a just right in order for the cells to function normally. And there can be several environmental influences such as lithium chloride somehow entering the body abnormally, which can affect cell processes. So here are our sources and resources, and we would like to thank the Medical College and Dr. Shala for providing us with the means of running our own experiments and learn more about this pathway. Thank you very much.
Thank you, Kettle Moraine.